Have you ever just stopped and thought about what it actually takes to keep a piece of a living being alive, growing, and thriving completely outside of a body? I mean, it sounds like science fiction, right? But it's actually the very real, highly meticulous science of animal cell culture. Because when we figure out how to cultivate cells on a massive scale, we literally unlock the power to test life-saving drugs, manufacture vital vaccines, and even reverse organ transplant rejection. So let's jump right into this explainer of Chapter 8 and discover exactly how we take biology out of the lab and turn it into life-saving medical marvels. To really understand where this incredible technology is today, we have to look back at where it all began, and that takes us to 1951. An African-American woman named Henrietta Lacks was diagnosed with terminal cervical cancer. During her treatment at Johns Hopkins, a doctor named George Gee took some cells from her cervix without her permission, mind you, but what he discovered was absolutely revolutionary. Unlike normal cells that just eventually die off in a lab, Henrietta Lacks' cells were immortal. They could be kept alive and grown indefinitely. These cells, which became known as the HeLa cell line, basically became the cornerstone of modern medicine. We're talking everything from testing the live polio vaccine to studying radiation effects. Her cells were widely commercialized and ended up generating millions and millions of dollars in profit for medical researchers. So what exactly are we doing when we culture these cells? Well, the formal definition of animal cell culture is the in vitro maintenance and proliferation of cells outside a living organism. In vitro literally just means taking place in a test tube or a culture dish, as opposed to in vivo, which means inside the living body. Honestly, it helps to think of these extracted cells as incredibly demanding VIP guests. They've been pulled out of the perfectly balanced, cozy environment of a living animal. So if they're going to survive and multiply, we have to bend over backwards to artificially supply them with absolutely perfect nutrients and growth conditions. Here is our roadmap for today's explainer. 1. Immortal cells and cell culture. 2. Nutrients and culture media. 3. Simulating the physical environment. 4. Types and viability of cells. And 5. Life-saving medical applications. All right, diving into Section 1, Nutrients and Culture Media. So our first major challenge is figuring out exactly what our demanding cellular VIPs actually need to eat to survive outside the body. Creating this perfect meal is what we call formulating the culture media. And the recipe? It is highly specific. Cells need a really complex mix: vitamins, amino acids to build proteins, glucose for energy, and inorganic salts. But honestly, the absolute most important ingredient on this list is serum. Fetal bovine serum is super common, and it is a complete biological powerhouse. Serum provides these crucial binding proteins like albumin and transferrin, which literally carry other molecules right into the cell. And even more importantly, serum gives us adhesion factors. See, most animal cells actually need to physically stick to a surface before they can even think about dividing. And serum gives them the biological glue they need to adhere to that culture dish. When researchers are picking a medium, they generally have to choose between two main categories. First, you have natural media. This consists entirely of natural biological substances, things like plasma, serum, and tissue extracts. On the flip side, we have artificial or synthetic media. This starts with a basic, synthesized basal medium, which the researchers then highly customize with specific supplements. So, depending on the exact cell line you are growing, you might toss in specific hormones, epidermal growth factors, or insulin-like growth factors to trigger very specific metabolic responses. Moving right along to Section 2, Simulating the Physical Environment. Now, simply feeding the cells, that isn't enough. We have to perfectly simulate the exact physical microenvironment they would normally experience inside a living, breathing animal. And one of the absolutely most critical physical factors here is osmolality. This is what maintains the integrity of the cell membrane itself. If the osmotic pressure outside the cell gets lower than the pressure inside, water rushes in, and the cell swells up and can burst. If it's higher, water rushes out and the cell shrinks and shrivels. So by incredibly carefully balancing the amounts of glucose and salt in the culture medium, commercial media are formulated to maintain a strict, precise final osmolality of right around 300 milliosmoles. Which brings us to another huge environmental hurdle: pH control. As these cells grow and metabolize, they generate waste products, and those waste products can drastically change the acidity of their little environment. To fight this, we use a bicarbonate buffering system inside the incubator, which relies on maintaining an atmosphere of 5 to 10% carbon dioxide. This whole system works on Le Chatelier's principle. Think of it like a chemical tug-of-war. If the acidity increases, which just means there are way too many hydrogen ions floating around in there, free bicarbonate ions immediately jump in, react with those extra hydrogen ions, and form carbonic acid. This shifts the reaction and perfectly instantly stabilizes the pH. But even with those amazing buffers doing their job, scientists need to know instantly if that pH balance starts to fail. Enter phenol red. This is a chemical that gets added to commercial media, and it acts as this brilliant visual alarm system. If the medium gets too acidic, the liquid turns yellow. If it goes the other way and becomes too alkaline, it turns pink. But when the environment is perfectly balanced at that optimal pH of exactly 7.4, the liquid is a bright, vibrant, healthy red. Okay, Section 3, Types and Viability of Cells. Now that we know how to feed and house these guys, let's talk about how we classify their lifespans and how we actually double-check if our fancy environmental setups are keeping them alive. Broadly speaking, there are two main types of cultures. A primary cell culture is exactly what it sounds like, obtained directly from host tissue. These cells are dissociated straight from a parental tissue and dropped into a container. But normal primary cells do eventually stop dividing. On the other side of the coin, we have continuous cell lines. Just like those HeLa cells we talked about earlier, these are immortalized cells that have undergone a transformation. They retain this incredible ability to just divide indefinitely, as long as we keep feeding them. The thing is, constantly maintaining living cells 24/7 is super expensive and highly vulnerable to things like bacterial contamination. So scientists use a process called cryopreservation to freeze them for later. But you can't just toss living cells into your kitchen freezer. The water inside them would instantly form ice crystals and completely shred the cell membranes. There is a precise recipe here. It involves adding cryoprotective agents, usually a mix of 10% DMSO and 90% serum. Then the cooling process has to be incredibly slow, taking them all the way down to -80°C. This slow pace lets the water safely escape the cells before they actually freeze. Then, when you need to wake them back up, you have to thaw them as rapidly as possible in a 37-degree water bath. That speed minimizes any chance of those lethal ice crystals forming during the warm-up. Throughout all of this feeding, culturing, freezing, and thawing, researchers have to constantly measure cell viability. That's just a fancy way of saying checking how many cells are actually alive versus dead. Why does this matter so much? Well, beyond just making sure your own culture is happy and healthy, viability assays are the main way we test the real-world impact of chemicals. We use them to see if a new pesticide is effective, to evaluate exactly how much cellular damage is caused by environmental toxins, and honestly, most importantly, to test the safety and the true potential of brand new pharmaceutical drugs. The absolutely most common way to do this check is the Trypan Blue exclusion assay, and it's wonderfully elegant. Trypan Blue is a specific dye that simply cannot penetrate the intact membrane of a healthy living cell. It just bounces right off. But if a cell is dead, its membrane is compromised, and that blue dye just floods right in. So, looking through a microscope, a researcher can instantly tell the difference. The clear cells are alive and well, and the ones stained bright blue, those are dead. And that brings us to Section 4, Life-Saving Medical Applications. All of this stuff, the meticulous lab work, mixing the perfect media, the strict temperature controls, running the viability assays, it all leads up to the ultimate payoff, and that payoff is real-world applications that are literally saving human lives. Animal cell culture has basically become this massive biological production house for incredibly important pharmaceutical proteins. For example, by using Chinese hamster ovary cells, or CHO cells, we can manufacture erythropoietin to treat severe anemia. We use them to produce factor VIII, which is a crucial blood-clotting glycoprotein that saves patients suffering from hemophilia A. CHO cells are even used to pump out TPA, tissue plasminogen activator, which dissolves blood clots and is an approved immediate life-saving treatment for patients actively experiencing a heart attack or a stroke. It's wild to think these tiny cell factories are doing all this. But one of the most vital, truly game-changing applications of this entire technology is the creation of monoclonal antibodies. Let's look at the mechanism of a therapeutic drug called OKT3. This is an immunosuppressant given intravenously to patients who are receiving a kidney, heart, or liver transplant. See, when a new organ is transplanted, the patient's own T cells recognize it as a foreign invader and start attacking it, causing acute organ rejection. OKT3 acts like a microscopic precision smart bomb. It hones in and binds directly to the CD3 receptor on those attacking T cells. This forces the T cell to become completely inert. It blocks its function and effectively reverses the acute rejection of the transplanted organ. And the best part? Once the therapy is over, normal T cell function returns. It is quite literally a lifesaver, and it's made possible entirely by animal cell culture. So, we've just seen how keeping a few cells alive in a lab can lead to breakthrough stroke treatments and massive organ transplant success. But as we wrap up this explainer, I think it's deeply fascinating to think all the way back to Henrietta Lacks. We started with her story, those cells taken way back in 1951 that sparked a total biological revolution and generated, frankly, immense pharmaceutical profits. And looking back at that origin story, it leaves us with a genuinely profound question, one that scientists and ethicists are still debating today. When an immortal cell line has the power to cure diseases and build entire medical empires, who really owns the rights to that biological breakthrough? Is it the patient whose body provided the tissue in the first place, or is it the scientist who figured out how to keep it alive? It's a heavy thought, and it's something that continues to shape the ethics of biotechnology right now. Keep exploring, keep questioning, and thank you so much for joining me on this explainer.